We will be talking tonight about stem cells and about the stroke. You might not realize that, but many people, especially aged population, dies due to the stroke. And when it comes to the disability, this is the leading cause of the disability of humans. And the stroke is a very complicated disease. Many of you know about it. Who doesn't, I will not go into detail anyway. It's too complex. But what is important that there is a certain degree of recovery. No matter how severe the stroke is, if you survive, you will get certain degree of the recovery. There is a machinery in our brain which helps us to regain the function which has been lost. So there is a spontaneous recreation, regaining of the function. However, we don't have any developed strategy or any therapeutic ways to help to regain this function in this recovery phase. Stem cells and the cells which I will describe right now could be used to improve this phase of the regeneration and improvement of the condition of the stroke patients. Now let's switch gear and talk about the stem cells. What are they? This is a very primitive cell which basically doesn't have any function except two. It could divide and give the origin to two similar daughter cells. So this is self-reproduction capacity of the cell. And if you put these cells under certain conditions, you can generate very specialized cells. Cells like muscle cells, like heart cells, like neurons, beta cells which produce insulin, etc., etc. During the development, when the zygote gives rise to the blastocyst, in the blastocyst there is an inner cell mass. And these cells are embryonic stem cells. Most often when we talk about stem cells, those are the cells which we mean. And they are responsible for all the other cells which are present in our body, more than 200 types of cells. They will give rise to all these cells. In the culture system, we can isolate these cells, put them into the dish, and expand them. By expansion, we are utilizing the property of the stem cells to be able to divide and generate new stem cells. And this expansion capacity gives us the tools to study in the culture system many cells, and to generate then from the different types of the cells for studying or for application in animal models. Today there are possibilities to use the postmitotic or already differentiated cells like skin cells, and then from that skin cells generate pluripotent stem cells. And this is the Nobel Prize winning discovery. In 2012, Nobel Prize to Yamanaka went for this discovery that he can make from skin cells pluripotent stem cells, so-called induced pluripotent stem cells. This is amazing. I mean, this is happening in the dish. You put the skin cells, you transfect that with certain viruses, certain genes, and in 2 weeks' time, in your view, under the microscope, you can observe how these cells are transformed into beautiful neurons. So what are the ways by which the stem cells can be applied to the brain or can be utilized in order to improve the function? Firstly, we might generate these neural stem cells in the culture system and transplant in the damaged brain with the hope that they will become neurons and they replace the neurons which died due to the ischemic stroke. Secondly, if we will learn how this system of neurogenesis, this production of new neurons, manipulates in the brain, what we can do, we can enhance this process by drugs or by different treatments and enhance the plasticity of the brain and support the recovery. And the last possibility is by manipulating the immune system, because as it has been shown in previous talk, there is activation of the immune cells in the brain, and these immune cells have very important function to the recovery and the plasticity. And then these stem cells can influence the properties of these immune cells, and through acting on the immune cells, we might be able to improve the recovery after the stroke. So this is the stroke damaged brain, and this is the area, this ventricular area, which is very close to the stroke-induced damage. And in 12 years ago, in 2002, we have published paper in the Nature Medicine when we looked what is happening with these cells. And those are the cells which are normally migrating to the olfactory bulb. But once the stroke damage occurs, what is happening that they start to migrate towards the damage. And once they enter this damaged area or adjacent to the damaged area, they will become neurons, and they become the neurons, the same type of neurons which died due to the stroke. Initially we thought that it was an acute event, but obviously it turned to be very long-lasting event, and for several weeks, several months, this process of the neurogenic response continues, and not only the young neurons but also mature neurons will be produced many, many months after the stroke. And here there are data from our publication where after 1 year animal got stroke, 1 year later we injected BRDU and labeled the newly formed neurons, and we can still see much less, but still production of new neurons. So there is kind of permanent switch of the mode, how the brain operates once it gets the trauma. And we believe that these cells are involved in some plasticity changes or rebuilding of the network, which could be contributing to the recovery after the stroke. In humans, there are data indicating that this is happening also in human brain, and there was last month there was a paper by Jonas Friesen showing with carbon-14 labeling that in the human brain after the stroke in the striatum there is production of new neurons. So hopefully these cells are also involved in the regeneration and plasticity changes, and we will be able to use this technology to improve the patient's condition. By using these cells, postdoc in my lab has transplanted them into the cortex and the striatum of the mice, and then studied how well they can pick up the food and eat the food with left and right hand. Normally there is no problem, but once they get the stroke, then there is a problem of getting the food because of the motor impairment of the limbs. And what happened that the animals which get transplantation, they can perform much better. So there was certainly functional effect on the behavior. Most importantly, as I told you, the neurons should be functional. And here you can see that these neurons, which have been grafted in these animals for many, many months, once you depolarize them, they can generate action potentials. This is electrical signals which neurons should generate if they are functional neurons. And again, we are facing situation where these cells are operating in the aged brain, and they do operate, because we have recently completed study with 2-year-old rats in the cortical damage. If you will implant, they will not only improve the performance in the behavior, but they will also decrease the size of the lesion. And by using this behavioral test, we can show that with the cells transplanted group, there is much better recovery as compared to the non-transplanted animals. And these are the aged animals, 2-year-old animals, which is good news for the stroke research, that this system is operating not only in the young, relatively young adult animals, but also in the aged animals. And now, what are the requirements? I mean, where are we from the clinical perspective? How should we direct our research to achieve the ultimate goal? First of all, it is necessary to define what is required from the stem cell-based approaches to be clinically competitive, and what risks to be patients are acceptable. Many clinical trials are studied before we get really clear functional effect in the animal models of the stroke. Unless we have that, we believe, there is too risky or too premature to bring this into the clinic. In the relevant animal models, we should have the proof of principle that how they work and that they work in order to move it to the clinic. And the biological mechanisms in this respect are very important. Many clinicians have the approach that as long as it works, I don't care how it works. What is the mechanism behind it? But we, as a basic researcher, are very concerned about the mechanism. For one hand, we want to know that this mechanism will ensure that it is long-lasting effect with minimal side effects. And secondly, with knowing the mechanism, we can always work on improvement first of its efficacy, and second, decrease of the possible side effects. And in the end, I would say that these efforts to bring something new in the clinic, it's not only our responsibility as the basic researchers, but clinicians should be involved on the other side. They should define what we need to do, what needs to be developed, what is the requirement for the patients from the stem cells could be. And we should move from both directions and meet somewhere in between in order to create something valuable which will benefit the patients and help them to improve their quality of life. Thank you very much. That's all.